Hello everyone. Today we are going to see about the microscope's parts. As in a previous lecture we discussed about the what is a microscope. As microscope, it is a device which is going to enlarge the image and it is used for magnifying the image. So different microscopes are going to be invented. It is going to be what we call that is a lens system is used in a microscope, which is going to be categorize the microscope into two types, a single microscope and your compound microscope. A single microscope is a device which is going to use with a single lens system. It is a single microscope. Again, a compound microscope, that is a microscope having the more than two lens system. Now we are today going to discuss about the microscope parts, that is it is a example of a compound microscope which is comprises lens system, that is a one than more than two lens system. Here we discuss one by one what are the parts of a microscope. Let's see. A microscope, it is a parts. A first part, that is a what we call it is here a eyepiece or a ocular lens. These are a eyepiece or ocular lens comprises two lenses, that is a it is called as a binocular because of a two lens. As a eyepiece they are going to be manufactured here. A eyepiece lens, it is comprises with a power that is called as a 10x or a 15x. After that we see here what is a head going to be monitor here ocular lens. Then we are going to see here what we call it is a nose piece. A nose piece, it is also called as revolving ocular here. Again here a nose piece contains three types of objective lenses. Objective lenses which are going to be in a different what we call that a magnification power. Magnification power of our objective lenses are like a 10x is there, 45x is there. Again there is a oil immersion objective. Oil immersion objective, it requires the oils which is going to encompass the what we call that is the refractive index of the medium. So after that below objective lens, that is there is a arm that is going to be here, we see here arm is holding for carrying our microscope from one place to the another. That is here arm is going to be hold, that is your body tube, your eyepiece and your mechanical stage. Next about the after arm we are going to see that is a here mechanical stage is there. It is going to be a stage clip incorporated with a stage clip. Mechanical stage is used for holding your specimen. Whenever we are going to do the any staining, any what we call that is culture, preparation of a smear, which things you have to be observed, it is have to be placed in this mechanical stage. A mechanical stage is supported with stage clips. A stage clip is going to be hold of slides a specimen in a proper position. So there are stage clips are provided. Now see here in a microscope mechanical stage is there. After that there is a knobs are there. There are two types of knobs, that is a coarse adjustment knob and here below that is a coarse adjustment knob. There is a presence of a small knob, that is a fine adjustment knob. These adjustment knobs are going to be used whenever you focus your specimen. First we have to, when we are going to handle the microscope, you have to use the coarse adjustment knob for focusing a specimen. After that specimen is once focused, then for a clarity of image you are going to use a fine adjustment knob. So there is a stage control knobs are there. Whenever stage controls of knob, these are going to be up and down. Your mechanical stage can be go through the your what we call the objective side. You can pull down, that is towards the your diaphragm size. After that below the stage there is a two important things are present, which is called here diaphragm. It is a circular ring which is controlled for a amount of light to be passed. See here diaphragm is there, it is a circular ring is there. Here is a clip, we are going to be what in a clockwise anti-clockwise directions. By using this diaphragm you can cut a light or you can take a more that is light to the specimen. Again there is a small hole in that your mechanical stage, it is called as a aperture. That is here a small hole is present which is going to what pass the light from the specimen, that is in a circular form. Then below the diaphragm there is a condenser is there. What is the function of a condenser? It is how to be concentrate the light towards your specimen which is going to be a pass. Below that condenser there is a illuminator, light source is present. Now see here, this is a light microscope, that is inbuilt light source is there, but in our daily basis, that is our laboratory, you have seen a compound microscope, that are a light microscope that we place towards the window side where sunlight is going to be come. In that microscope there is a no what is light source, illuminator is not present, but there is a presence of a circular mirror which is going to be what? Reflect the sunlight that is going to be passed towards your condenser. Again here light source is there, it is an inbuilt light source is there. Whenever you are going to operate this microscope, you have to only switch on, switch off. After switch on, a light source is emitted from this which is going to be passed towards the condenser here. After that, it is going to pass through the aperture your specimen, then it is again reflected to the objective. From objective it is going to be enlarged with the help of a eyepiece. Now see here, there is a brightness adjustment knob is there. After that, this it is called as what is a base. Depending upon your base, the whole microscope assembly is going to be what comprises here. After that, a light switch is button is there, you have to use a base. Then there is a arm is carrying a handle also is called, apertures, stage clip, objective lens, nose piece, your ocular lens. Now we are going to see how the what we call that is a working of a microscope is going to be a form. Whenever we are going to outer look in this, what is here what we call that is a stage, a specimen comprises a stained slide, then it is a how to going to be work, how the image is going to be enlarged. We see here, whenever we switch on this light pass through this condenser. After that, condenser function, that is a concentration of light, then it is concentrated light is passed through your specimen, that is from through the stage, aperture is present which is going to be your specimen. From the specimen which light to be reflected, it is going to be a comprises from your objective lens. Now the image from this area, it is called as a real image. Still there is a not, we are not going to get an enlarged image, but when the from the objective lens image reflected their rays and these are go through the eyepiece, when we are go through our eyes, we got an enlarged image. Means here magnification is going to be increased by the power of your multiplication of your eyepiece and your nose piece power. Suppose my eyepiece is a power that is a 10x and I am going to use a objective lens of a 10x, then my image is going to be enlarged by 100 times. Means whenever we are going to see from the ocular lens, you are going to observe the virtual image, that is not a real image, it is enlarged due to the magnification power of this microscope. Similarly, if you are going to use the eyepiece with what we call that is a 10x is eyepiece, then your objective lens is 40, then your magnification power would be a 450. Similarly, when you are going to use a 10x with a oil immersion, that is a oil immersion, how the magnification power of a 100x, then your image is going to be enlarged with a 1000 times. So these are a simple description of a microscope parts. See, we are going to use, this is a compound microscope. There are many microscopes are going to be invented. They are going to be a design for a practical purpose by the need of a researcher. For example, there are microscopes such as a phase contrast microscope is there, fluorescence microscope is there, electron microscope, in that a scanning electron microscope and your transmission electron microscope, these are going to be used. Similarly, compound microscope have the version of a dark field. Now see here, a compound microscope, what we are going to use, it is a type of a bright field microscopy, because here your specimen is going to be what, stained here, and the background is bright. Whenever we are going to observe to the compound microscope, it is also said that it is a version of a bright field microscopy. So thank you.